Hello everyone and welcome to ITLS Academy Empower the Youth. So today in this lecture we will going to study about one technique, that is Agarose Gel electrophoresis. So Agarose Gel electrophoresis is a electrophoretic technique that is used in different laboratory for experimental purposes. In this particular lecture, we will going to see what is Agarose Gel electrophoresis, what is the principle working of Agarose Gel electrophoresis, as well as we will see the applications in different industries and what are their disadvantages and advantages of this technique. So let us understand what is Agarose Gel electrophoresis, but before understanding the definition of this, let us understand what is electrophoretic technique is. So electrophoretic technique is a separation technique that is based on the movement of charged particle in an electric field. Electrophoresis is an analytical method of separating charged particles based on their relative mobilities in an electric field. So whenever we are talking about this technique, that is electrophoresis, so electrophoresis is nothing but it is a separation technique, separation of different type of particles that is based on the movement of the charged particle, how they are moving in an electric field. And electrophoresis is a analytical method of separating the charged particle based on their mobilities, means on the relative mobilities in an electric field, means how they are moving in an electric field. Depending upon their relative mobilities, their separation take place. So it is an analytical method. As you can see in this image, here is a plus sign and here is a negative sign. So we all know that one side it is anode and another side it is a cathode, right? So anode and cathode means whenever a positive charge is there, whenever there is a negative charge, so in between a positive and a negative there is always a electric field. Then in between these two charges, how these particles are moving, how these charged particles are moving with their relative mobilities in an electric field. So depending upon that, this electrophoresis means the separation take place. Now let's move further and let us understand what is an Agarose gel electrophoresis. So Agarose gel electrophoresis is a method of gel electrophoresis that is used in biochemistry, molecular biology, genetics and clinical chemistry to separate a mixed population of macromolecules such as DNA, RNA or protein in a matrix of agarose. So what is agarose gel electrophoresis? So agarose gel electrophoresis, it is a method of gel electrophoresis, and one of the method of gel electrophoresis that is mainly used in laboratories of biochemistry, in labs of molecular biology, in the labs of genetics, clinical chemistry. Here they separate a mixed population of macromolecules, not micro, macro, that a little that have as a, that is bigger in size, like such as DNA or RNA or molecules in a matrix of agarose. So they separate this type of population in the matrix of agarose. Agarose, what is agarose? Agarose is a natural linear polymer that is extracted from the seaweed that form a gel matrix by hydrogen bonding when heated in a buffer or allowed to cool. So what is agarose? Agarose, it is a natural liner polymer. What is an agarose? It is a natural liner polymer that is extracted from a seaweed. From where they are extracted? From the seaweed, and that form a gel matrix. And how they are forming a gel matrix? By a hydrogen bonding when in the heat, when they are heated in a buffer and further they are allowed to cool. So they are the most popular medium for the separation of moderate and a large sized nuclei acid and have a very wide range of separation. So they are the most popular medium. One of the most popular medium is agarose, and this medium is generally used for the separation of moderate or a large size nucleic acid and have a wide range of separation, or in simple word you can say they separate a mixed population of macromolecule like DNA, RNA, protein, nucleic acid, right? And it is a natural linear polymer. Sometimes in interviews or in MCQs also they ask you that what is agarose, so you have to tell that agarose is a natural linear polymer that is basically extracted from the seaweed, and that forms a gel matrix by hydrogen bonding. When they form this gel matrix by a hydrogen bonding, when they are further heated in a buffer and further they are allowed to cool. Now moving further, that this is how the agarose gel electrophoresis look like. So this is a gel tank, as you can say this part, this is a gel tank with agarose in it. One side they have a positive sign, positively charged, and second side is it is here is a negative charge, and all these are connected with a power supply. So in this way the setup of agarose gel electrophoresis is there in the laboratory. So whenever you go to the laboratory, whenever you visit your respective laboratories, so you can see this type of setup is there, which is used for the separation of macromolecules. Now, what is the principle behind this agarose gel electrophoresis? So, gel electrophoresis separates DNA fragments by size in a solid support medium such as agarose gel. Sample DNA are piped into a sample well, followed by the application of an electric current, which causes the negatively charged DNA to migrate towards the anode, that is a positive ends. The rate of migration is proportional to the size molecular fragments move quickly and wind up with the bottom of the gel. So so how they are working? So the gel electrophoresis, what they do the separation. They separate the DNA fragment. How they separate the DNA fragment? So they generally separate the DNA fragment by a size in a solid support medium. Which type of solid support medium? Agarose gel. So the sample means the DNA, they are pipetted into the sample well. While they are pipetted into the sample well, that is the agarose gel, and they are followed by electric current. So one side it is a negatively charged and another side they are a positively charged. So they are they have the two types of current, positive and negative, and which causes the negatively charged DNA to migrate towards the anodal, that is a positive size end. So the rate of migration, so whenever it comes to the migration, so the rate of migration is proportional to the size. Smaller fragments they move more quickly to the bottom of the gel. So depending upon the size, they travel, means the particles that are small in size, they move more quickly towards the bottom of the gel, and the particles that are larger in size, so they move less quickly, means they move after the all the small molecules migrate. So here you can simply understand, here also the size play a vital role in the movement, means the larger size is moving more quickly than the smaller one. The smaller one is moving more quickly than the larger one. Then now let's see the second point. DNA is visualized by including in a gel in an intercalating dye, etherium bromide. DNA fragment is taken up by the dye as they migrate through the gel. Illumination with the ultraviolet light causes the intercalated dye to fluorescence. The larger fragment fluoresces more intensely, although more quickly. So DNA fragment take up the dye as they migrate through the gel. Illumination with the ultraviolet light causes the intercalated dye to fluorescence. So the larger fragment fluoresces more intensely, although each of the fragment of a single class of a molecule is present in the equimolar proportion. The smaller fragment include the less mass of DNA, take up the less dye and therefore fluorescence less intensely. A ladder set of DNA fragment of a known size can be run simultaneously and used to estimate the size of the other unknown fragment. Now comes the requirement or the instrumentation of Agarose Gel electrophoresis. Means what are the equipments or requirements of Agarose Gel electrophoresis? So the equipment and the supplies necessary for conducting Agarose Gel electrophoresis are relatively simple and include an electrophoresis chamber and a power supply. As you can see in this diagram, uh, this one, so they need a chamber and they need a power supply. Okay, so the gel casting tray that we have seen in the diagrams, which are available in a variety of sizes, and they are composed of UV transparent plastic. So the gel casting tray, that's this tray is composed of they are present in the various sizes, whichever sizes they is needed, they are present in that sizes, and they are composed of UV transparent plastic. Then the open end of the tray are closed with the tape while the gel is being casted and removed prior to the electrophoresis. Now comes the sample comb. So sample comb around which the molded medium is poured to form a sample well in the gel. Then comes the electrophoresis buffer. So electrophoresis buffer, it is basically of two type, that is a tris acetate, that is EDTA, or a tris borate, EDTA TBE. So it is present, they basically have two types of buffer. Now comes the loading buffer. So loading buffer which contains something dense, that is glycol, to allow the sample to fall into the sample wells, and one or two tracking dyes which migrate in the gel and allow the visual monitoring or so, or how far the electrophoresis has been proceed. So now comes the staining. DNA molecule are easily visualized under an ultraviolet lamp when the electrophoresis is the presence of the electron extrinsic no, fluoroethodium bromide. Alternatively, nucleic acid can be stained after electrophoretic separation by soaking the gel in the solution of the ethodium bromide. When intercalculated into the double-stranded DNA, fluorescence of this molecule increases greatly. It is also possible to detect DNA with an extensive fluor 1 animalimo 8 naphthalene or sulfonate transluminator, an electroviolet light box which is used to visualize the strained DNA in the gel. So as you can see this diagram, the DNA sample is loaded here, and this is a plastic tray, this is a Agarose gel, and this is how the sample is loaded into this. And this, as you can say, this is a comb where you are loading the your sample. At one end it is a negative, and where is another end it is a positive, means at one end it is a cathode, whereas in the another end it is a anode. And you are giving a power supply, basically 60 to 150 watt power supply is given. And yes, this is how the Agarose gel electrophoresis are there. So what is the concentration of the agarose gel electrophoresis? Means what is the concentration of agarose gel? So if we talk about the concentration of agarose gel, then the percentage of agarose gel used depends on the size of fragment that is to be resolved. The concentration of agarose is referred to as a percentage of agarose to volume of buffer, that is weight by volume, and agarose gel are normally in the range of 0 to 3%. Again, this is very important. You must be aware of the volume of the agarose gel that is used. So how much agarose is used? Generally 2 to 3 per 0.2 to 3% agarose gel is used, that is weight by volume. And the concentration of agarose gel is generally refers to the percentage of agarose to volume. The lower the concentration of agarose, the faster the DNA fragments migrates. So whenever the concentration of agarose is lower, so the more faster the DNA fragments they get migrated. So in general term, if we talk about, if the aim is to separate the large DNA fragment, a low concentration of agarose should be used. So if your aim is to separate the large DNA fragments, let's suppose your aim is to separate the large DNA fragment, so it is always recommended to use the low concentration of agarose. And if the aim is to separate the small DNA fragment, then high concentration of agarose is recommended. Why? Because the lower the concentration of agarose, the faster the DNA migrates. Right? So this is the case whenever you want to separate a large DNA fragment, then you should always use a low concentration. And if you want to separate a small DNA fragment, then you should always use a high concentration of agarose gel. So this is the concentration. So you should always be aware of this concentration, because in examination or in various interviews they may ask you the range or the concentration. So you must be aware of this term, that is 0.2% to 3%. This is very important. And of course this point is again very important. If you want, you can take the screenshot of this thing. I'm just making it more visible to you. So take a screenshot so that you can easily memorize these terms. And why sometimes in interviews or in MCQ, in various examination, they ask you this, why? So the answer is here: the lower the concentration of agarose, the faster the DNA fragments migrate. Okay, now moving further, to prepare agarose gel, to prepare a gel, agarose powder is mixed with the electrophoresis buffer. Means how you make a agarose gel? So you take agarose powder and you just mix this powder with the electrophoresis, and after mixing the with the electrophoresis buffer to a desired concentration, means whatever concentration you need, you mix with the desired concentration, and then you have to heat it in a microwave or microwave oven to melt it. So after after mixing it in a buffer, you keep it in a microwave oven to make it melted or heat for heating purposes. And yes, then further it is the ethidium bromide is added to the gel. Then what you add? You generally add the ethidium bromide is added to the gel. And what is the final concentration? The final concentration would be 0.5 to facilitate the visualization of DNA after electrophoresis. Then you keep it for the cooling purpose. Then after cooling, the solution is about 60 degrees centigrade. It is then further poured into the casting tray that we have already discussed, a tray that is composed of UV plastic material. Okay, the is poured casting tray containing a sample comb, and you just and allowed to solidify at the room temperature. So you pour that solution, whole thing of solution, into the tray, and keep it at a room temperature. Why? You just to solidify. After the gel has been solidified, the comb is removed. Once it gets solidified, you just remove the comb, and taking care not to rip the bottom of the well. You have to take it out very attentively, so that there should be no rip of the bottom of the well. The gel is still in the plastic tray. Then it is inserted horizontally into the electrophoresis chamber, and you have to cover it with the buffer. The sample containing DNA mixed with the loading buffer, and then they are pipetted into the sample well. The lid and the power leads are placed and on the apparatus, and the current is applied. The current flow can be confirmed by observing the bubbles coming off the electrodes, that is negative, and the positive, that is anode and cathode. Then what they do, then the DNA, they migrate towards the positive electrode. So the DNA, where they first migrate, they migrated towards the positive electrode, which is usually colored red. In view of this negative charge, the distance DNA has migrated in the gel, they can be judged by visually monitoring the migration of the tracking trace, like bromophenol blue and xenoline and cannalol dye. So this is how the concentration of agarose gel take place, and this is the steps, then how this experiment is being performed. Now let's quickly move to the applications of agarose gel electrophoresis. So agarose gel electrophoresis is routinely used for the method of separation of protein, RNA and DNA. Estimation of the size of DNA molecule. Then they are used in the analysis of PCR products. PCR products, for example, molecular genetics diagnosis or genetic fingerprinting. Then the separation of restricted genomic DNA prior to the southern analysis, or RNA prior to the northern analysis, means southern blotting and northern blotting. Then the agarose gel electrophoresis is widely employed to estimate the size of DNA fragment after digesting with the restriction enzyme, that is restriction mapping of cloned DNA. Agarose gel electrophoresis, they are commonly used to resolve the circular DNA, okay, with different supercoiling topology, and they are involved to resolve the fragment that differ due to the DNA synthesis. In addition to provide an excellent medium from the fragment size analysis, agarose gel allow purification of DNA fragment. So this one is again very important. Apart from separation or of macromolecules, it is also involved in the purification of DNA fragment. So since the purification of DNA fragment size separated in a agarose gel, then it is necessary for a number molecular techniques such as cloning. It is vital to be able to purify fragment of interest from the gel. So if you see, so apart from the separation techniques, it is also involved in the purification of different DNA fragment. So as the last point says, that since the purification of DNA fragment size separated, In an agarose gel, so it is necessary for a number of molecular techniques such as cloning. So it is also involved in cloning of DNA. So it is vital to be able to purify the fragment of interest from the gel. So it has a great advantages, or you can say applications where the cloning of DNA take place. Now understand what is the advantages and disadvantages of agarose gel electrophoresis. So for the most application, only a single component agarose gel is needed, and with no polymerization catalyst are required. So this is an advantages, means no polymerization catalyst is involved. So you are just saving a capital cost. Why? Because only a single component, that is agarose, is needed. So therefore agarose gel are simple and rapid to prepare. Why? Because agarose gel are very simple, and they are rapid to prepare, means you can easily make the agarose gel. So the gel is easily poured and does not denature the sample. This is again a great advantage, means you can simply pour the gel, and after pouring the gel, they does not get denatured the samples. The sample can also be recovered, means if you want to recover your sample, so you can easily recover your sample through the agarose gel electrophoresis. So these are certain advantages of agarose gel electrophoresis. Now we have talked about the advantages. Everything has advantages as well as disadvantages. So if we talk about the disadvantages of agarose gel electrophoresis, so the gel can melt during the electrophoresis, means whenever it's an electrophoresis is conducted, so the gel get melted. So this is one of the disadvantages. The buffer can become exhausted, means the buffer that you have used, so it can become exhausted. Different form of genetic material may run in an unpredictable form. So this is again a disadvantages. So that's bring to the end of the lecture. I hope you have understood the lecture. If you have any doubt, any queries related to the lecture, or anything you have in the previous lectures, so you can just simply leave your queries in the comment section below. In the next lecture I will be answering the queries. And yes student, follow ITLS Academy on Twitter, Instagram, Facebook, YouTube, WhatsApp, LinkedIn. You can get our app, that is ITLS Academy, on Google Play as well. And you can just subscribe our channel, YouTube channel, in the name by the name of ITLS Academy, because in this channel you may get all the videos related to different topics. And you can visit our website, that is www.itlsacademy.com. This is our helpline number, that is 7080833450. You can simply call on this number and get updated. And again students, thank you for watching. Please do like, share and subscribe, and don't forget to press the bell icon to get more updated. Thank you students.